Hi everyone, my name is Anusha Reddy Govindu, and today I'm going to present a fascinating study titled Cell Shape Affects the Bacterial Colony Growth Under Physical Confinement. This work was originally done by M. Sri Padmanabh and colleagues, and I have chosen it because it beautifully connects microbiology, physical environments, and cell morphology, which also ties into what we have been discussing in this course. Here's what I'll be covering today. I'll start with some background to explain why bacterial cell shape matters. Then, I'll talk about the main goals of the study, walk you through the methods they use, and share some of the key results. Finally, I'll summarize the conclusions and highlight how this ties into across topics and microbial ecology and cell behavior under stress. So, why does the cell shape matters and what is cell morphology? Cell morphology refers to the shape, size, and structure of a cell. It usually describes the physical appearance of bacterial cells. The bacterial shape is not just a random trait. It plays a major role in how colonies grow and interact with their environment. For instance, rod-shaped bacteria like E. coli or Bacillus tend to align neatly with each other and physical barriers. This is similar to pencils aligning in a box. On the other hand, speckle-shaped bacteria like Staphylococcus are more randomly arranged. These structural differences influence how colonies expand, how fast they grow, and how well they pack under tight spaces, especially in the places like the gut. See, the shapes will be like cocci, which is spherical, bacilli, rod shaped, spirella, spiral shaped, and their arrangement will be like chains, clusters, space. Understanding cell morphology helps to identify and classify microorganisms under a microscope. See, as I told you earlier, rod-shaped bacteria, they tend to line up with each other and the walls, whereas the spherical ones, they don't align with the same way. This alignment affects the direction of the colony expansion, speed of the growth, and packing and crowding. So, what were the researchers trying to find out? Essentially, they wanted to explore how bacteria grow when confined in tight 3D spaces like the mucous layer of the gut. To mimic this environment, they created 3D microenvironments that can simulate the physical confinement. Methods. They like preparation of 3D growth media, hydrogel matrix made from the carbopol C980, and the concentration should be 0.5% for low confinement and 0.85% for high confinement, and the pH adjustment should be neutralized to 7.4 with NaOH. Viscoelasticity testing rheology is to mimic mucus stiffness, and the characterization of gel matrix, pore size analysis, data analysis, matrix stability. Uh, to stimulate the confined environments, the researchers use the hydrogels with tunable pore sizes, especially essentially controlling how much space the bacteria have to grow. They use the confocal microscopy and also the softwares like MATLAB to analyze the pore structure and confirm the matrix stayed stable over time. There's no significant change in the rheological properties over 24 hours at 30 degrees Celsius. And the next part is the bacterial cultures and assays. They like bacterial strain isolation and the culture. They isolated 7 gut bacteria from red floor beetles, which were identified via 16S RNA sequencing. Um, they monitored growth through optical density and used dyes and markers to distinguish between the live and dead cells as well as to track stress responses. See, the optical density, it should be measured every 10 minutes for 16 hours. The cell viability checked using the propylene iodide for dead cells, calcium or GFP signal for live cells, DNA stress monitored using HGFE and SURNA M3 reporters. Uh, Colony analysis and imaging, colony shape studied using the confocal Z-stack imaging, image analysis, and also comparison of rod versus spherical colonies. They also ran simulations using cellmorphology, a platform that allowed them to model how rod and spear-shaped bacteria behave under confinement, focusing on the nutrient access and growth direction. So the discussion of the results. Now let's get into it. First, they found that the different physical properties like the stiffness of gel significantly impacted how the bacteria grew. They confirmed this using the absorbance and confocal imaging. The physical properties of the mucosal layer are diverse and are altered by the factors such as a change in diet, infection, inflammation, and enzymatic degradation of fibers by microbes. Different gut derived microbial strains from red flower beetles were imaged in bright field, pseudocolored for enhanced contrast. They create an in vitro 3D growth medium that offers a granular and internally porous microenvironment, enabling the bacterial growth under physical confinement. By densely packing highly swollen polymeric hydrogel granules beyond the jamming threshold. See here the 3D growth media mimicking physical properties of the mucus. The properties of microgels are tunable based on the mass, percentage of hydrogel granules relative to the liquid LB. Here they show two kinds of gels, soft and low confinement. As I told you earlier, for soft and low confinement 0.5 and stiff and high 0.85. Rheological measurements are done with three kinds of methods. They confirm that the cell shape matters. Rod-shaped bacteria like E. coli tended to form long directional colonies, while spherical bacteria grew into compact clusters. A change in confinement alters bacterial population growth. Bacterial growth monitoring in 3D gels by measuring absorbance, and here B, bacterial growth curves over 16 hours of all tested strains with levels of confinement. And here the C, this one, increased confinement differentiates bacterial species into two distinct growth response groups as shown by the relative biomass ratios at 16 hours. Here, next, OD measurements reliably reflect actual microbial load in 3D media as validated by confocal microscopy based cell counts, with both methods showing comparable relative biomass ratios after 16 hours of growth. And here from you can see from E to G, these three, motility doesn't provide a significant growth advantage under confinement. This was tested with motile and non-motile E. coli strains. Bacterial morphology determines colony architecture and competitive outcomes. Agent-based simulations show that high aspect ratio cells grow directionally along the parent cell axis, while low aspect ratio cells grow more randomly in 3D space. Rod-shaped E. coli, high aspect ratio, here in B, form elongated colonies under confinement by pushing their progeny outwards. Next, see here treatment with A22, which disrupts Ameribian round E. coli cells, results in spherical colonies. D. Naturally spherical E. fecalis exhibits growth patterns similar to low aspect ratio cells in simulations, and A22 treated E. coli. Next, a factor analysis confirms significant morphological differences between colonies of high and low aspect ratio cells in both simulations. Shaken liquid culture growth of rod-shaped and spherical E. coli shows no significant difference in growth rates. Relative biomass ratios indicates that the rod-shaped E. coli are less impacted by confinement than spherical cells, maintaining ratios closer to 1. Next, 3D co-culture assays show high aspect ratio cells. Example, motile E. hormicki non-motile E. coli outgrow low aspect ratio cells. Competitive co-cultures between two high aspect ratio strains do not show significant enrichment of either strain, suggesting similar fitness. Biological replicates within less than 130 to 400 cells per condition. Genomic DNA analysis from co-culture experiments suppose that the high aspect ratio bacteria are more resilient to increased confinement compared to low aspect ratio strains. Single cell morphology enables physical confinement to selectively favor elongated colony architecture. Growth assess indicates that P. sanguineus performs at par with rod-shaped bacteria across two different degrees of confinement. Measurements of the aspect ratios of bacterial cell bodies for all 10 strains used in the study measured from the samples in the late exponential phase. Microbial growth under confinement is described using a phase space that relates relative biomass prediction changes to single cell morphology. So, finally, in competitive assays, rod-shaped bacteria outperform the spherical ones under confinement, but when two rod-shaped strains were grown together, neither dominated, suggesting that the cell shape was a key factor in competition, but not the only one. So, to sum up, this study clearly demonstrates that the bacterial morphology strongly influences colony structure and growth under confinement. Rod-shaped, high aspect ratio bacteria outperform the spherical ones, like the spherical low aspect ratio bacteria under physical confinement. Rod-shaped cells form elongated colonies that expand directionally, enabling better nutrient access. Spherical shells grow into compact rounded colonies, limiting nutrient diffusion and growth. Agent-based simulations predict and match experimental results. The cell shape governs colony structure. Changing cell shape. Example, E. coli made spherical using the A22 drug, which changes colony growth patterns. Motile and non-motile strains of the same species of E. coli perform similarly under confinement. Nutrient conditions. Shape-based growth advantage holds true across both rich and minimal nutrient environments. In competitive assays, high aspect ratio bacteria dominate under high confinement, reducing the relative abundance of spherical bacteria. No dominance observed when two rod-shaped species are co-cultured. Rod-shaped bacteria can better exploit directional growth and nutrient access in tight spaces. These findings are not only relevant for understanding the gut microbiomes, but also for developing better probiotics and also the microbial therapies. And so, this study clearly demonstrates that the bacterial morphology strongly influence colony structure and growth under confinement, whereas the rod-shaped ones can better exploit directional growth and nutrient access in tight spaces. See, the rod-shaped ones are the high aspect ratio ones, which outperform the low aspect ones. The rod-shaped ones form the elongated ones, but the spherical shells grow into the compact rounded ones. And motile and non-motile strains of the same species perform similarly under confinement. These findings are not only relevant for understanding gut microbiomes, but also for developing the better probiotics and also microbial therapies. So, thank you for listening, and I'd be happy to have any questions.